Good morning, everyone, and happy Friday. Before I get started, are there any questions regarding the course? Okay, so I have news for you. Your exam one is ready, and Ansley is working on your study guides. So once she's finished, which should be early next week, she will send an email around and let you know where to find the study guide for exam one. And exam one will take place during the class time. It will be on Monday, so well, not next Monday, but the Monday after next. And you will have from 9:30 to 10:20 a.m. to complete it. And if you need accommodations, please be sure to go through Student Accommodation Services office to request the appropriate accommodation. And if you have sent an email to me already, I got you covered. All right, so let me share my screen here and get to the lecture. So quiz 2 is due today. Don't forget to take quiz 2. I looked at how many people took it, have completed it by yesterday, and it was not a lot. It was almost half, which is, do it early. Just do it early, and if you remember, you have 2 tries, and if you don't know much answers on the first try, schedule office hours with me. Discuss the confusing concepts. Maybe you don't understand the question, so schedule office hours with me or Ansley, and we'll get to the bottom of it. And this is the benefit of taking quizzes early. Quiz 3 opens tomorrow or at midnight tonight, and it is due next Friday. And finally, the participation assignment is due today, and it's a Friday kind of participation assignment. So some of the people, I'm asking you to submit your favorite biology related meme or joke, and so far it's been pretty good. If you want to brighten your mood before the weekend, just go to the discussion and browse through the jokes and memes already posted in there. It's pretty good, and submit your own. Contribute to the Friday fun. All right. So before diving into today's topic and the schedule, I wanted to spend some time on the previous topic, which was DNA structure and function. I finished last lecture with the mutations, with agents of mutation and how mutations affect DNA and what are the consequences of that. So the examples I gave was sickle cell disease as a result of the substitution mutation within the DNA, and it causes the hemoglobin, the red blood cells to stick together because of the aggregation of a hemoglobin, the protein in the cell. I mentioned that the crops that we have today, of the food that we eat today, is very much mutated. It's not in its original form. So in agriculture, mutations are widely used. But I wanted to also mention about something about the viruses, which are the agents of in introduction of DNA into a foreign cell. So viruses themselves can bring new genes into cells. And so it is used in agriculture now. The virus in agriculture industry, people, scientists use viruses as a means of delivery of the gene to a particular locus in the DNA, to a particular location in the DNA. And but I will not talk about that. I'm just mentioning it just for general education. But what I do want to talk about is what happens when a virus enters the cell, how a virus makes the cell do what the virus needs to do to survive. So in the picture here, you see a variety of viral shapes. We have a tobacco mosaic virus, which attacks the tobacco plant. We have this spaceship kind of virus, looking virus. We have rotavirus, which, as far as I remember, causes pretty bad diarrhea, and herpes virus, which causes outbreak on the skin. So all have different shapes, but All of them have the same mechanism, how they enter the cells and how they make the cells to produce viral DNA or RNA and viral protein codes in order for the viruses to survive. Okay. So viruses are smaller and simpler than cells, and technically, a reminder to you that viruses are not alive. It is technically not a cell. It is an agent with a protein coat inside of which some DNA or RNA is contained. So DNA or RNA is technically the genetic information within a virus, but this agent, which is not even alive, can do pretty serious damage to the host, be it a plant or be it a human organism. So viruses are smaller than cells and they don't have a nucleus, they don't have an organelle, they don't have a cell membrane, they have a protein coat. So examples of viruses are smallpox virus, influenza virus, common cold, rabies, polio, warts are all caused by viruses. So viruses, they hijack the transcription and translation in cells. The information encoded in the viral genes, information is encoded in viral genes, but viruses don't have the equipment to express their genes on their own. So viruses must infect cells in order to replicate themselves. So look at the virus and look at the true living cell. So in order to replicate, the virus would have to undergo transcription and translation. So virus contains only genetic information. So in order to reproduce, it would need to replicate the genetic information and replicate the protein code, but it doesn't have the ribosomes. It doesn't have the organelle to actually do the synthesis of the new proteins. It doesn't have an organelle. It doesn't have the enzymes to replicate the DNA. It doesn't have the enzymes to synthesize RNA. So what it does, it infects the host cell and uses the translation and transcription and translation mechanisms inside the host cell in order to replicate. Did somebody unmute to ask the question? Natalie, go ahead.
So my question is, if if viruses aren't really alive, can they die? Because we do a lot. When we inject vaccines, can we kill them even though they're not alive?
Oh, yes, we can. Yes. So if when we get a vaccine, and I will touch on it later, when we get a vaccine, we train our immune system to recognize the type of protein code for a particular virus, and that makes our white blood cells attack the viruses and digest them. Remember, I covered I mentioned the phagocytosis a couple of lectures ago, and I gave the white blood cells as an example.
Montague, come on.
Okay.
Please stop.
I'm gonna mute Ava and her dog. So the white blood cells, they perform phagocytosis in our bodies. So the white blood cells, they can be trained to recognize the particular foreign proteins, and vaccines are doing exactly that. They're training the white blood cells to recognize the protein codes on the viruses. And if the white blood cell circulates our system and it finds the viral protein, it immediately engulfs it and digests it. So
okay.
This is how viruses are killed.
So we just kind of rip them apart so they can't function anymore.
Yep, that's exactly right.
Okay, thank you.
No problem. Okay, there's a thing in the chat. Do vaccines only contain the protein codes? That's a good question. Various vaccines differ. So there are RNA vaccines, there are vaccines that contain only the viral proteins. So RNA vaccine, as far as I know, some vaccines contain the genetic material itself, and they train the body to destroy the genetic material, and some vaccines train the body to destroy the protein code. But I'm not really a vaccine specialist, so I'm not sure to what extent I can answer that question fully.
An example. I listened to an epidemiologist a couple times, and one of the COVID vaccines, for examples, it literally just it rips apart the protein coat and just lets the RNA and DNA, disperse, and then it's just dead. If you rip off that, the iconic like Corona, why it's called, it has the special outside.
Mhm.
It just rips that apart, and then once it's ripped apart, it can't penetrate a cell and create more viruses.
All right. Thank you for that addition, Dylan. Very, very informative. Okay. So, moving on. So how do viruses how do viruses enter the cells? They first attach to the cells. So on the outside of the cell, this is the rough depiction of the cell membrane, okay? And remember that within a cell membrane, we have some proteins embedded in it. And some of the proteins are called receptors, and viruses, on the outside of the protein coat of the virus, they also have some proteins called receptors. And if the receptor protein on the coat of the virus can match precisely with the receptor protein on the cell, the virus will attach to this receptor protein and through it enter inside of the cell. So viral proteins grab onto proteins on the host cell, and that's how they come inside the cells. Not all of the cells are susceptible to all of the viruses. For instance, the HIV virus, the human immunodeficiency virus, it can only infect the white blood cells. It cannot infect the skin cells. It cannot infect various other cells in the body. The only cells it can infect is the white blood cells. That's why, like, if you shake hands with the HIV positive person, you will not catch the HIV virus. So viruses, after they enter the cells, they inject their genes into the host cells. So viral DNA or RNA enters the cell, and the cell begins to synthesize the viral nucleic acids and proteins. So here's our virus that has entered the cell and has disintegrated. The protein coat has disintegrated, and the viral DNA enters the cell and undergoes, if the virus has DNA, then through the cell's process of transcription, the messenger RNA is created from the viral DNA, and then this messenger RNA attaches to the ribosomes and produces proteins that are needed for the viral protein coat. Okay, and if the virus has RNA instead of the DNA, then transcription is not going on. The process jumps straight to translation. But the point is, the whole cell replicates viral DNA or RNA and creates a protein coat that is needed for the virus. So after this process repeats over and over and over and over again. So eventually the host cell becomes full of viruses. And once it's full of viruses, the host cell bursts, and the viruses exit the host cell and infect the neighboring host cells. So an example I can give you that demonstrates this process, that you can feel this process is happening, is a common cold. So during the common cold, you feel such symptoms as runny nose, inflamed throat, like these burning nasal passages. So this is because the viruses are ruining the cells in the throat, in the nose, and so on. So you're feeling the symptoms because the cells are getting destroyed, okay? But we get better after a cold. We get better within a week from a cold. So what is happening? And what is happening is our immune system kind of wakes up, it attacks the viruses, and eventually the cells that got destroyed they regenerate pretty quickly. They regenerate within a week. And once the cells have regenerated, the cold symptoms disappear. And again, it happens usually within a week. So vaccines are the most effective way to prevent viral infections. And as we've mentioned so far, vaccines teach our immune system to recognize a virus and eliminate it from the body before many cells are infected. And this is our most potent weapon against many viral diseases. So some vaccines provide immunity just for 1 year. For instance, a flu shot, it's effective only for 1 year. And the reason for that is because viruses mutate very rapidly. So a vaccine that was effective for a particular flu strain last year may not be effective for this year's particular flu strain. And usually the type of vaccine that is used in US relies on the type of vaccine that is used in Australia, because in Australia, the flu season starts half a year earlier compared to US flu season. So other vaccines like polio vaccine, which I'm hoping all of you received, I'm sure all of you received, is effective for 18 years. And actually there's two types of polio vaccines. One polio vaccine contains the live polio virus, and another vaccine contains just the protein, or not the live ones, the dead ones. And another vaccine contains the protein. So in countries like Ukraine, where I'm from, we received the type of vaccine with the dead virus, which is considered more dangerous, but it gives you the lifelong immunity. And in US, I believe the most widespread is the type of vaccine that gives you immunity for 18 years. The measles vaccines protect you for the whole life once you receive the 2 shots of it. And vaccines have been instrumental in eliminating some of the diseases. So for instance, the smallpox, which was bad disease. First, it disfigured your appearance. And second, many people died from it because it not only affected your appearance from the outside, but also from the inside. And it did some serious damage to your organs. So smallpox has vanished because of the widespread vaccinations. Okay, that's weird. And polio is nearly gone because of the intense efforts to vaccinate people in the countries where polio is widespread, and there is not enough means to vaccinate a lot of people. But some diseases are now re-emerging. For instance, measles started to re-emerge particularly in the communities who believe that vaccinations cause autism, which absolutely bogus and not true. But yeah, these beliefs are having real toll on the public health.
I was gonna say, you said earlier we probably all have the vaccine. I was gonna say most of us probably got the vaccine before anti-vaxxers even became a movement that was well known at all.
Well, they have been active for a while. I mean, since
Just
the
quietly though.
70s. Like the movement itself emerged in the 70s. I mean, people started having these beliefs in the 70s, but yeah. But it's always kind of there's always a group of people who will deny what's good for you and deny the mainstream views. So sometimes it's harmless, but in the case with the vaccinations, it's not so harmless. It causes the damage to herd immunity.
Yeah, especially I know, I was sorry I keep interrupting, but I've read stories forever ago about polio rising, and I just thought, it was a real shame. We thought we almost eradicated the disease.
Yeah. And if you don't know what polio is, it kind of
Look up the iron lung. That's what you should look up.
the iron lung. Yeah, it's a serious one, and if you end up surviving the polio, there's a good chance that you get crippled. I mean, you cannot
It's
move
terrible.
normally. Yeah. So, but I feel like I'm scaremongering. Anyway, so there's another way to deal with viruses, which is much less effective compared to the vaccinations. And it's called the antiviral drugs. So, and the way the antiviral drugs work, they slow viral replications. So they can stop viruses from binding to cells or block them from hijacking the transcription and translation in cells. But they're not very effective for a long time. And the reason why they're not very effective, these drugs are not effective for a long time, is that viruses mutate rapidly and they are so diverse genetically that it is challenging to produce drugs with long-term effectiveness. So lots of effort was put into HIV treatments, to drugs that would slow HIV virus replication. And over time, these drugs have become less effective, so new drugs have been developed, and the development of the new drugs is driven by mutations in the virus. Okay, and that's it for the viruses. Does anybody have any questions before I move on to DNA replication, binary fission, and mitosis? Go ahead, Madeline.
I have another question. So what you just said about how vaccines are not as effective when
No.
a virus mutates.
Antiviral drugs are not as effective as vaccines. Vaccines are still more effective.
Oh, okay. So what's an antiviral drug?
Ooh, antiviral drug. I can't remember.
Like an antibiotic?
It's not an antibiotic. No, antibiotic and antiviral drugs are two separate things. And by the way, a common misconception, like if you get viral pneumonia, it's a very widespread misconception that you should take antibiotics. Antibiotics work on bacteria. Antibiotics work on cells and not on viruses. Okay, so do not take antibiotics if you have a viral disease, unless the doctor prescribes it. But sometimes people just treat themselves and think they know better. Anyway, so the example of the antiviral drug, I'm blanking on the one. There was a very famous one, which was also mentioned against there was a famous drug that was for HIV, and it's caused big damage to the body, and they even made a movie about it with Matthew McConaughey. I'm blanking on the name of the drug. There's a Z.
I know what you're talking about, and I have
AZT.
no idea what it's called.
Carrie just mentioned AZT. That's right. AZT is an example of the antiviral drug. And Madeline also mentioned Tamiflu is an antiviral drug. So Tamiflu is, well, it's in the name what it treats. It was used to treat the swine flu, I believe, when it was widespread. Did I answer the question, Madeline? Or did we collectively answer your question, Madeline?
Yeah, I guess so. Thank you.
All right, any other questions? Okay, moving on. So our next topic is about DNA replication, binary fission, and mitosis. And binary fission and mitosis are both ways of cell multiplication, cell division. So all cells divide. No living organism can reproduce without cell division. Cell divide in our bodies every second, every millisecond, as we speak. So unicellular organisms divide in order to reproduce, and cell division produces continuous supply of replacement cells in our multicellular organisms. In multicellular organisms. So if we did not have a way to replace the shed cells, we would wear away. For instance, in our skin cells are constantly dividing, and in fact, the top layer of the skin is dead cells. The living cells are underneath the dead cells. So, and we're constantly shedding the dead cells. And if we lost the dead cells and the new ones did not divide, we would just end up with no skin. Okay, and a fun fact, every 35 days, we have a completely new layer of skin, because that's the turnover time for the skin. That's how long it takes for the skin cells to completely shed and the new skins skin cells to be produced. the cells not only divide and replicate, but they also die. Skin cells die, and I just mentioned that the top layer of our skin is the dead skin cells, and the development of an organism requires not only cell division but also cell death. If you remember from grade school, this picture of the embryo development, of a human embryo, even like at some point in the development of the human embryo, we all have tail, like the resemblance of a tail. But when we're born, we don't have a tail. So the tail itself, the cells in the tail, they die, and their death is programmed. And this programmed cell death, there is even a term for it. It's called apoptosis. So apoptosis is a programmed cell death, and it's a process that can carve out distinctive structures during development. So during human embryo development, it gets rid of our tail. But here is another example of the apoptosis. A chick and a duck, when they're embryos at embryonic stage, they both have webbed feet. So the webs in the feet, they are preserved once in the baby duck and in the adult ducks, but the the cells in the webs of the chick embryo, they die or undergo apoptosis, and it results in a chick not having webbed feet like a duck, but they start out with the same webbed feet as embryos. So there are three types of cell division in all cells. Not all of the cells have the same. So okay, I'm making it more complicated. I'm just going to leave it at that. There are three types of cell division. Binary fission, and it is only prokaryotes. Yes, only prokaryotes undergo binary fission. And prokaryotes, again, are unicellular organisms that do not have a nucleus and cell-bound, the membrane-bound organelles. The second type of cell division is mitosis. Mitosis happens in eukaryotes. So we have mitosis happening in our bodies as we speak. Our skin cells divide. The division of a zygote, which is a fertilized egg, is also undergoes via mitosis. And the third type of cell division is meiosis. And meiosis is the cell division via meiosis happens when we produce, during production of the sex cells, or half of the total amount of parent genes are packaged into the resulting cells, the cells that have undergone meiosis. So I'll go into more details of what is the difference between mitosis and meiosis later on. But in general terms, during mitosis, the cells end up with the exactly same number of chromosomes in the end. And during meiosis, after cell division, cells end up with only half of the chromosomes as in the parent cells. So before the cell divides, it has to create a copy of its genome. And genome is the collection of all genes in the cell, or a DNA molecule. Okay, before a cell divides, it needs to make sure that it splits. It has some DNA to give to a daughter cell. So the entire genome is replicated so that each new cell gets one complete copy of the DNA. And it's a short slide. There's nothing much to tell there. How does DNA replicate? So DNA, similar to synthesis of the RNA, DNA is replicated with the help of the proteins called enzymes, specifically DNA polymerase. So it's not important how these enzymes are called, but it's important to remember that DNA replication is aided by enzymes. Enzymes help to replicate the DNA. Okay, so the first stage. Enzymes called helicases unwind the DNA. Again, it's not important to remember the name. It's important that enzymes unwind the DNA. So here's our helicase, the unzipping enzyme, and it goes along a DNA molecule and unzips it, unwinds it, because in its natural state, in its steady state, the DNA molecule is a double winded double helix that's kind of that has a spiral shape, and the rungs on the letter of the DNA are joined together by hydrogen bonds. So this purple blob, the helicase enzyme, the unzipping enzyme, it unwinds the DNA and breaks the hydrogen bonds between the nitrogenous bases. Now, after the DNA is unwound and unzipped, the DNA polymerases, they synthesize new DNA strands. So one DNA polymerase approaches one strand and starts synthesizing the complementary DNA strand, and another DNA polymerase, it approaches the opposite strand and starts synthesizing the complementary DNA strand. And a reminder, the complementary means that if the guanine is read by the DNA polymerase, the cytosine is going to be synthesized. If adenine is read by the polymerase, then the thymine is going to be synthesized, and so on. And finally, enzymes called ligases join short strands into long strands. And I'm not showing it here. I think there is a slide. I'm going to skip ahead. So when do ligases come into play? The DNA replication can start at several locations at once. Okay, it doesn't, the DNA, the enzyme that unzips the DNA, there's not just one enzyme. Several enzymes can approach the DNA strand at several locations and unzip it at several different locations. And so the ligases, once the DNA has been synthesized, the ligases kind of join these several newly synthesized DNA together at various locations, okay? And this synthesis of the new DNA at various locations along the original DNA strand, it just speeds up the DNA synthesis. The sole purpose of that is to make DNA replication faster. So now back to this slide. The DNA replication is considered to be semi-conservative. So what it means that the cell keeps the original parental DNA each time replication takes place, and keeps while producing new DNA at the same time. So in the image here, the dark blue DNA strand is the parental strand, and the light blue strand is the newly synthesized strand. So when the DNA is replicated, the bottom line is, when DNA is replicated in the new DNA, in the newly created DNA, half of it will be the parental DNA, parental cell DNA. All right, and I just covered basically this, that DNA replication begins simultaneously at multiple spots along the chromosome, and it speeds up the DNA replication. And after, the way that this continuous strand of the new DNA is created is with the help of the enzymes called ligases, which fuse these separate DNA strands, newly created DNA strands together. Okay, so my next slide is about binary fission. And so are there any questions about this DNA replication process?
Would you mind going back over the DNA being semi-conservative again, please?
Yeah, so semi-conservative, meaning in the new DNA, when DNA replicates from one DNA molecule, when one DNA molecule becomes two DNA molecules, the new DNA molecule is not going to be completely new synthesized DNA. In the new DNA molecule, one strand of the DNA will be from the parent, will be old, and the other's parent cell. And the other strand of the DNA will be the newly synthesized DNA, the DNA that was synthesized just now, just during the process of this replication. Did that make this better, Lauren?
Yes, thank you.
Okay. Other questions about this seemingly complicated process of the DNA replication? Going once. Going twice.
Could you go over the third step about how long strands join into short strands?
Okay, so the step number three. Step number three is not really, it's not depicted in this image very well, so I skipped to this image, okay? So step number three is more visible here. So we have light blue strands, which are newly synthesized DNA, dark blue strands, which are old DNA, parent DNA. So we see that there is a chunk of the newly synthesized DNA over here, and the chunk of the newly synthesized DNA over here. Eventually, they will come together, but there will be no bond with them. So step number three, the enzyme called ligases join the short strands into long strands. So, oops, wrong way. The ligases join this bit to this bit once they're synthesized all the way to each other. Did that clarify it?
I got you. Thank you.
Okay, great. More questions? Going once, going twice. And to the binary fission we go. So prokaryotes divide by binary fission. And among the prokaryotes, who belongs to prokaryotes? Bacteria and archaea. They reproduce asexually, meaning they don't undergo meiosis, and the only way they can reproduce is through binary fission, or completely replicating its DNA, creating identical DNAs to the parent cell. Well, almost identical, because there's always a possibility of a mistake or a mutation. But if there's no mutations and no mistakes, if the parent cell of the bacteria will always be identical to the daughter cell of the bacteria. But if you've heard about antibiotic resistant bacteria, then you might wonder, well, how does that happen? Like, how do they come to be? If the binary fission produces two identical cells with the two identical DNA, how can bacteria become resistant to the drugs, to the antibiotics? And the answer to that is through mutation. So there's always a small probability of mutation in the DNA. And if the mutation is beneficial for the survival of the bacteria in its environment, then that's going to be the DNA that's going to be replicated over and over and over again. So if we have a bacterial infection and we are treated with antibiotics, and suddenly there was an unfortunate mutation, unfortunate for us, mutation in the bacterial DNA, but very fortunate mutation for the bacterial DNA, that provided resistance, that provided the means for survival of that bacterium in the antibiotic-rich environment. That kind of bacteria is going to replicate and take over, like continue to survive, basically. It will continue to divide and creating more and more bacterial cells that are resistant to antibiotics. Okay. So here are the stages of this binary fission, and it is pretty simple. There is nothing too complicated to it, unlike multiple stages of mitosis and meiosis. But basically, in the first stage, or rather in the second stage, for some reason here. The DNA replicates and attaches to the cell membrane in the slightly different parts of the membrane. And DNA replication takes place in the same mechanism that I discussed in the previous couple of slides. So once the DNA is replicated and attached to different parts of the membrane within the cell, the membrane grows in between, in the middle of the cell, and eventually, because the membrane grows in the middle of the cell, separating the two DNA molecules, the cells become two, one cell becomes two cells. And this is the gist of the binary fission. It's basically replicate the DNA, attach the DNA to two opposite cell location, and then grow a membrane in between the cell. Voila, you have two different cells. Binary fission. Any questions about binary fission? Okay, if not, we're going to move to complicated eukaryotes and complicated eukaryotic division and replication process. Well, replication is the same. Mechanism of replication is the same for prokaryotes and eukaryotes, DNA replication that is. But eukaryotes, the cell division in the eukaryotes undergoes much more stages. So first stage is in replication is that DNA winds up very tightly around the chromosome. And you can see like the two, compare visually two cells. This is cell number one, this is cell number two. So if the cell number one does not prepare to divide, the cell number two prepares to divide. Now in cell number one, you can see the DNA is very loosely packed, and the analogy that is given in the book is the loose yarn. Whereas in the cell that prepares to divide, the DNA is analogous to a neatly coiled yarn. And in fact, the common depiction of the chromosome as like X's, it's really the depiction of the chromosome before the cell, when the cell is fixing to divide, okay? Because the chromosome, when the chromosome is depicted as an X, it's basically a depiction of the DNA that is very tightly wound. It's a depiction of the replicated DNA that is very tightly wound into a chromosome. Looking through the chat, no questions in chat. Nancy's got it. Okay. Eukaryotic chromosomes are visible during cell division. So as I just mentioned, when DNA is loosely packed, you cannot really see the chromosomes. You can just see genetic material. But just before the cell starts to divide, it packs up its DNA into a tightly wound chromosomes. It packs up its replicated DNA into these tightly wound chromosomes, and this is when we can see them through a microscope. We can see the actual X-shaped chromosomes in the microscope. Wow, I'm starting to speak words not really well. So, the steps. First, there's this loosely packed genetic material, and this is kind of a zoomed in version of the loosely packed genetic material, so unwound chromosome. It replicates the DNA within the chromosome. And who remembers what a chromosome is? How is it different from the DNA?
Is it the DNA from both parents combined? Just a guess.
No. So how does the chromosome differ from a DNA? This is a chromosome. How is it different from a DNA? No. Come on, guys. So Salvador, chromosomes have DNA packed in. Exactly right. So chromosome is the combination of the DNA molecule and the proteins that around which the DNA molecules are wound up. Okay. So a chromosome contains the DNA molecule plus some proteins which help DNA molecule kind of pack well. So here's an unwound chromosome. The proteins are not depicted. I think the only DNA molecule is depicted. So DNA replicates, creating two chromosomes, and they're joined together in the middle with a structure called the centromere. And just before the replication, before cell division, DNA condenses around, it winds up tighter around the proteins contained on the chromosome. And this is when we start seeing an X-shaped chromosome in the microscope, if we were to look at the cell under the microscope. Okay, so this is the X is the replicated chromosome. So this chromosome, this chromatid, I'm sorry, this chromatid is identical to this chromatid because they originated from one unwound chromosome. So chromatid is 1/2 of the replicated chromosome. All right, eukaryotic chromosomes are highly condensed DNAs. Each chromosome consists of two identical sister chromatids, one for each copy of the replicated DNA. So this is kind of more convenient to explain on this slide. So this chromatid is exactly the same as this chromatid. And we have some sort of alleles here. And so this region is identical to this region is identical to this region is identical to this region, and so on. So the next part I was planning to go over is mitosis, but that's pretty big start for this is a pretty ambitious start for 4 minutes. So I'm going to stop here and open the floor to the questions.
Can you hear me?
Yep.
Okay. So I guess I still don't really understand, like, chromosomes have DNA. Like, why is that why it's different from DNA? Like, I don't really get that. Like, I know it has DNA, but like, why is that?
Let me think of an analogy here.
I was gonna say it's like organized DNA. I mean, you're gonna come up with a way better analogy, but to simplify, it's almost just like organized DNA.
Well, organized DNA, it's a good attempt.
It's always organized. I mean, it's always organized, but it's even more organized now.
Right. So let's say you have a tree, okay? A tree has leaves. So when leaves is a part of a tree. So in this analogy, DNA is leaves and tree is a chromosome. So DNA is part of the chromosome.
Okay.
But not the only part. So let's say a tree consists of leaves and trunk. A chromosome consists of DNA and proteins, which help pack the DNA, which
Okay.
help kind of make the DNA occupy less space. So did that clear it up, Carrie?
Yeah, kind of. Okay, thank you.
No problem. Any other questions?
Professor.
Yeah, go ahead, Anil.
This one more has to do with the final exam. For the final exam, is it going to be stuff? Because I don't know when are we going to continue on lectures next week and include that information for that Monday exam? in the not this Monday, but like the next Monday after that? Or are we stopping like a couple of lectures before to study, or how's that go?
We're stopping a couple of lectures. Like the exam will not contain, I don't think it will contain the information from this lecture even. So to study, you would need to focus on lectures prior to this one. But it will all be in the study guide. The study guide will be, I think it will be very helpful to you, and it will be posted early next week, okay? So if you want to save time, just go by the study guide.
Okay, thank you.
All right, guys, it's 10:20. Don't forget the participation exercise for today and look over other submissions. They're quite good. All right, have a good weekend, and I'll see you all on Monday.